Thank you, Neil, for the very nice introduction. And it's a pleasure to be here. Now, I understand I'm talking to mostly physicists, right? But are there any biophysicists, biologists in the audience? Okay. I will try to communicate to the physicists why you should at least pay attention to what is happening in the area of molecular biology, biochemistry, biophysics, things like that. Because we're beginning to approach where it might be a dream that you can understand life processes and ultimately the cell really in terms of physics. And by understand terms of physics, I mean quantitative equations that predict things, that you can go out and test them and see if it's true or not. If you think of most of biology, there are very few, very hardcore quantitative predictions. If you go into psychiatry, there are none, virtually none. And so as we understand more deeply than the feeling among physicists, if you understand it, then you can calculate something, you can predict something. Okay, so with that, I'm going to talk about one type of molecular motor. So the cells use little molecular machines to traffic material back and forth. As an example, if you're a neuron, the longest neuron in your body goes from your hip to your knee, one single cell. And you can imagine in this neuron, you've got to transport material. The directions of how to make proteins and things come from the nucleus, but you've got to transform along long distances. In this cartoon of the neuron, you have the cell body on the left-hand side, and you've got to transport these things all the way to the distal axon side. And then these axons are then form synapses with other neurons in a highly connected part of the brain. One neuron could be talking to as many as 10,000 other cells. What do they transport? Well, they transport chemical cargoes. They transport machinery that actually makes ATP, because these are heavily energy-intensive cells, and you've got to make the local energy over these long distances. So there are two types of motors typically in a cell. One is called kinesin, which goes from the cell body to the end, and another is called dynein, and you're going to hear about dynein, which actually takes material from the distant end of the axon and transport it back to the cell. And you might think, well, why are you transporting materials to the other side? I thought neurons, they get molecular inputs from the dendrites. They decide to fire. They fire voltage pulses. So what is this business about transporting molecules back? And the point is that neurons and cells in general, have many chemical signals that say if you're attached and things are happy, you can proceed. And there are many danger signals when things are not happy. For example, as an example, we started this work in the early 2000s, looking at the transport of a molecule called nerve growth factor back to the cell body. If you don't get nerve growth factor from the environment of the distal axon, the cell thinks there must be something wrong, and it commits suicide. So it's a very, very important transport of this. Now, this molecular motor called dynein is a very big complex motor. It has 1.5 megadaltons, that's to say 1.5 million hydrogen masses worth of material. It's got a lot of complex molecules hooked up to it. And in the end, it's transporting a little vesicle called an endosome, and these are the dimensions of the endosome. So let me play you, first of all, there are various movie cartoons of, for example, kinesin. You've probably seen these, and it goes like this, it goes like this, and goes like this. This is utter nonsense, which I hope to convince you of. There's similar things with dynactin. This is a more realistic version. It's an animation made by Ron Vale, who is a pioneer in both kinesin and dynein. I think he discovered dynein. And so these things that you see here are the two legs of this motor protein. So these are the two legs, and let's see if they walk to music. And, and at this point here, it decides where to step next, and it's now searching to where to step. It finds out where to step, it lands it, locks it in place, and in this cartoon, once it locks it in place, that yellow and red linker arm then swings down and pulls the vesicle. So that's the cartoon image, how this works, but you can see it doesn't make physical predictions. And so before I begin, I want to tell you about a philosophy I had of all what I do in science, and it was stimulated by the great American philosopher of the 20th century. If in case you're wondering who it is, he's a baseball player named Yogi Berra. He played for the New York Yankees. He said many wise things, but the thing he said is you can observe a lot by just watching. He also said if you come to a fork in the road, take it. Or no one goes there anymore, it's too popular. You know, little funny things. And as a corollary, you can see more if you continue to watch for a long time. So this is the idea. We want to watch what's happening in live cells of this motor. So here's a quick outline. I'm going to tell you how we do it. We developed some improved optical probes, and then we watch in a live cell how this motor neuron transports its cargo. Now, all the hard work is done by the cell. We just got the cell to swallow up what it thinks is an important message, then it wants to take it back to the cell nucleus. So it puts on the motors, the attachment molecules, everything else, and then off it goes. You just have to convince the cell, take me back to this body. And I'll outline very briefly and not in detail the quantitative mechanical model. And then I'm going to go on to something which you may or may not know about. There's an exciting new field emerging called non-equilibrium statistical physics. And so let me start with the long-term molecular tracking. So we want to use a probe. These probes have been around for a couple decades. They're called rare earth upconverting nanoparticles. And when I got back to Stanford, I said, well, I'm not a material scientist, so I'll get some of my friends who do make these probes to give them to me, and I know what type of biology I want. And so I asked Paul Avasados to supply me with these, and he supplied them. And during that time, these optical probes are made starting with a crystal of sodium yttrium fluoride. And in that, this is optically inactive, but you can put optically active rare earth elements like ytterbium and erbium. But you're only putting in 20% of ytterbium. You put in that because photons, these things are only one unfilled shell, so it's just essentially a very complex, hyperfine type things at two levels. And they exchange energy amongst themselves, but they can resonantly transfer energy to this erbium. And so it's a two-step excitation. And so it's called upconverting because you shine in infrared light and you get out visible light in the green and red wavelengths. And so this has a lot of advantages. It's in the infrared, so you're not using visible light. Visible light is known to do bad things, create reactive ion species, all sorts of stuff. Cells don't autofluoresce when illuminated by anything longer than, let's say, 750, 800 nanometers. Unlike quantum dots, they don't blink and eventually photobleach. So this figure compares, for example, a fluorescent dye, a fluorescent protein, which doesn't last very long at all. Fluorescent dyes have gotten much better, but they too photobleach. And these are quantum dots, another order of magnitude or two longer lifetime, but they blink. And that was very annoying in the experiments we were doing in 2002, 2005. All right. So now the particles they supplied us with turned out not to work. They didn't know that, unbeknownst to them, what they thought were single particles were clusters of particles. You've probably seen molecular SEM pictures of these beautiful things in arrays. Those pictures are made because there's an organic molecule called oleic acid. It's used in the synthesis process. So when you lay them down, these oleic acid molecules, which you don't image, make them self-organize into these beautiful arrays. So what happened was they were much dimmer than expected. And so finally I threw in the towel and said, I think we're going to have to learn how to make these ourselves. And so the way we made them, my approach was to use a trick that's well known in the semiconductor industry. If you have a single crystal of gallium arsenide, and then through molecular beam epitaxy, you can actually grow some mixture of gallium arsenide and aluminum. And the atoms fall down and they assume the lattice spacing of the substrate, and you can grow anything you want with very high quality material. So this epitaxial growth, so I said, can we use epitaxial growth? Now the way you make these particles is in wet pot chemistry. So the idea was very simple. You start with a little seed crystal, 5 nanometers in diameter, and you put in next time you put ingredients that have the right mixture. And what we found is that when we do this, we can put in all optically active elements in any ratio we wanted. And so suddenly that means that because of the sheer concentration of optical things, you're up by a factor of 5. We also found that in attempts to make these particles water-soluble, the canonical wisdom is you have to strip off the organic oleic acid, use very harsh things like BF4 or hydrochloric acid. And so we're looking at TEM pictures of these particles. I was noticing, what are those little thingies on the edges? And one of the materials was very experienced in material science said, oh, that's probably damage. But if you have a little damage point, that could be a quench site. So I said, you forget about getting rid of the oleic acid, put something on top of it, squish it down, adds another nanometer or fraction of a nanometer of overhead. But if your particles are 25 nanometers in diameter, 30 nanometers, a 5 nanometer core doesn't really take away much. And then adding another half nanometer of stuff doesn't take away much. And so, and the upshot is we were able to make particles that were perhaps 100 to 200 times brighter at low intensity, suitable for animal in vitro imaging. And at the much higher laser intensities where they begin to saturate, they were still about 10 times brighter than anything existed before per unit volume. And so for that, we were off to the races. So it was just a simple idea taken from a different field. Instead of vacuum molecular beam epitaxy, it also works in wet pot chemistry. So the idea is we would take immature neurons, and we would induce them to grow through, if this was actually a commercial microchannel plate, and how do you induce them to grow? Well, you give them chemical signals and say, this is where you want to grow. Initially, many years ago, 2 decades ago, we used nerve growth factor, really expensive, but you can use really cheap substitutes, and it works. And so what you do is, in this microchannel plate that's nearly a millimeter in length, you can sprinkle on these probes, and here's a photograph of these optical probes, the upconverting nanoparticles. You're shining infrared light, they show green, and you can watch them move. And so you can actually take movies of the transport, and you can watch them for tens of minutes, and the cell doesn't die. And so this is an example of very early, many years ago, like half a dozen years ago, of this vesicle, which has this 1 nanoparticle, and it's going down, and as a function of time, you see the seconds ticking off. Sometimes it gets a little confused, goes backward, we don't know why. Sometimes it changes tracks, and when it changes tracks, it might too go backwards, and we don't know why yet. But in tracking this, you can actually plot what's going on. And the spatial resolution is a couple of nanometers, because you're finding the centroid of the fluorescence. Okay, so that's the technology. And so then we said, okay, now we're going to track them. And we're going to take video pictures of dot moving, and we can do it at different temperatures. Neurons like to be at body temperature, 37 degrees C, but room temperature, roughly 22 degrees C, which is where all the data, virtually all the data was taken of previous experiments. So we said, we can do it at 22 degrees, 30 degrees, 37 degrees. As you go to higher temperature, it walks faster because you're getting, I'll explain why in a second, but, and then what you can do is you can write a program that decides in its noisy data, when does it take a step? You don't want a human to decide. And from that, you ask the program to say, how big a step has it taken and how long a period of time between steps. And so the first thing we notice is if you look at the so-called dwell time, the time between the steps, at room temperature, there's a rise and a fall. And as you go to a higher temperature, there's a rise and a fall, and even higher temperature, a quicker rise, a quicker fall. And as soon as you see anything that's a rise and a fall, you know that the kinetics cannot be described by a single rate constant. So a rate constant, let's say if you excite an atom to an excited state and it has a certain lifetime, it'd be described by a simple rate, the lifetime probability of it coming back down. And you all know that that's a single exponential decay. So it starts at a high level and just decays. But here it starts at 0, rises and falls. And so you can play around with what might that be. But what we found that you can fit these curves, the black line fit, to a convolution of there's a simple rate constant of rate K1. And after that step is taken, then it has to go to another rate constant, K2. And so the total probability distribution in time is just the convolution of those two. And you can take that convolution, and these black curves turn out to just fit to 2 rate constants, and they happen to be equal. So just phenomenology so far, okay? So that's what we have. And you can show that with these 3 data points that the rate constants are consistent with a thermally activated process. All right, so that's what we learned. And so then you go back and figure out, well, if you have this molecule, this very complex molecule, the unit of energy is ATP. And the way you harvest the energy from these molecules is you take adenosine triphosphate and you break a phosphate bond, and you make adenosine diphosphate, ADP, and then there's a pi attached to it. Then the pi might leave, the unit of fuel lands on a molecule. It immediately hydrolyzes to this very quick electron transfer process, and then it loses the phosphate group, it loses ATP, and it goes into the empty state. APO in biology has said ATP is not there anymore. So these are the steps of any hydrolysis of ATP. These are the fundamental steps. But let's look at the rates. These have been measured before. Most of them have been measured. The time it takes ATP to actually bind onto this molecule, done in vitro experiments, allows us to estimate what the time of binding time would be at cellular concentrations. The initial experiments were done at very low concentrations, micromolar concentrations, to make it so slow that you can actually see single steps. But in biology, you don't have that. You're working at cellular concentrations, 3 orders of magnitude higher, and the binding time has been, if you just use those measurements, is about a millisecond. The hydrolysis is an electron transfer process. It's super, super fast, much less than a millisecond. It also turns out that in previous literature, they measured if you had ADP on the molecule at room temperature, how long does it take to thermally desorb? And that was measured to be less than a millisecond at room temperature. That leaves only one process that was unmeasured, and then how long does it take that phosphate group to leave the binding site? And so by default, that had to be it, and it's thermal desorption, so that would follow an Arrhenius activation energy. And so now we say, okay, there are two rate constants, K1, K2. They're both thermally desorbed. And so that's what we have. Now we look at the molecular structure of the business end of this molecule. This is a very, very complicated, it's a single amino acid strand that just goes winding around. And in that, there's people over several decades, 3 decades, studying this molecule, and they find there are 4 binding sites of ATP. But only 2 of them seem to involve with stepping. The others seem to gauge something on and off. And so we said, well, look, there are 2 rate constants, they're thermally desorbed. Is it possible that binding on the known sites in this cleft between, it's called, these are acronyms, but it's AAA1 and AAA3, could it be that you need hydrolysis in both these sites, and it has to be sequential? This was against the prevailing model suggested by Ron Vale in a landmark 2005 paper where he felt there was only one site. Okay. So then you're immediately in trouble because you're now going against the prevailing model of what's going on. And traditionally, that means the publication is delayed for about 2 to 3 years as a minimum, especially if you're not a molecular biologist. Okay, so we were stuck in COVID. Couldn't really take any data anymore. So what do? You think about, you read the literature, you think about your data, and working with Sam Peng, the graduate postdoc on this, we reviewed all the literature and said, what is the evidence that there was only 1 ATP versus 2? And we found that there was no compelling evidence that it had to be 1 ATP, not 2, except one notable thing, and that is when they looked at the dwell time between the time it took to make the next step, they saw a single rate constant. So this was the Ron Vale original proposed model, 2006. So it looks like a single exponential decay. But more subsequent experiments later on in the last 5 or 10 years, they begin to see a little nibble here, but nothing like this, which is our data. And so I'm friends with Ron Vale because he used optical tweezers in all of his single molecule experiments. And so I helped him with that, and he's a great guy. And so I said, well, and also talking with a postdoc at the time, Stefan Niekamp, now he's a graduate student, and he said, well, how did you analyze this? And he said, well, we decided how to bin it, and then after we bin it, we fit something. And said, well, why don't you do it in a different way? Use a program to decide how long it took before it made a step, but don't do any binning. And so you have many, many more data points and just fit directly to that data and see what you get. And after 2 days, what he found was reanalyzing, that's the original data that was published, reanalyzing it, he writes back and says, you know what, the best fit are 2 decay constants, K1 and K2, and they're equal. Now that may not look like a good fit because there's this hole here, but the limited data, so we did some simulations based on their data of what would happen, and that's just limited data. So the upshot of this work was, this more recent work says, yeah, there seemed to be a rise time and a fall time, 2 rate constants seemingly equal. Now the verdict's not in since then, and since we, in the last couple of years, other people have got improved measurements. They now all see a rise and a fall, but some of them don't see equal rate constants. So that's yet to be worked out. Okay, so now we're here, we have 2 rate constants, we think the active sites, and so we want to develop a quantitative model, much more quantitative than this cartoon movie. And the first thing we want, remember, we can see the time between steps, but we can also measure the steps. And again, in the original work of dynein in the Vale cell paper, you have distribution of steps. This histogram distribution is going forward in the correct direction, and this is the motor stepping backward. And of course, when it doesn't take a step, it doesn't take a step. There was a little confusion in the literature. Those dotted lines are 8 nanometers. Why 8 nanometers? You remember this motor taking the feet. It can be bind in a certain position on the microtubule, and the distance between the binding sites is 8 nanometers. But there's a simple thing. If you have 2 feet and you step 8 nanometers, and the other guy hasn't stepped, and this is my vesicle, how far is the vesicle moved? 4. And there was actually confusion in the literature. They think that the minimum step size should be 8. But that's okay. There's 4, okay? Point 1. Point 2 is our data corresponds to their data, but we're doing it in a live cell in vitro. We don't know how many motors there are. And so if you pick out the very slowest stepping, because our time resolution was marginal, if we say, it appears to be dynein, it appears to have the same step distribution as the in vitro experiments. In vitro is good because you put exactly what you want there. And so it tells you that the reconstituted motor in a test tube, in vitro means in glass. So in an artificial experiment, they've put in most of the essential ingredients that recapitulate what happens in live cells, and it tells us that we're seeing dynein, okay? Okay, so those are the step size distributions at 22 degrees, 30 degrees, and 37 degrees. So I said, well, let's do a fit to the data. And so the fit I wanted was a Gaussian distribution. And you're looking at this and saying, that's a Gaussian, well, come on, that's not a fit. But you have to, as an experimentalist, you have to be sensitive of what you can see and what you can't see. What you can't see because of the noise of our data and the noise of all the other data, in vitro data, is you don't see 0 steps because nothing moves. But you're unable to discern 4 nanometer steps. All right? So anyway, and there was something else puzzling. If you do this fit and say, the stuff that you can't see is because your step size resolution is not good enough and you don't measure 0, you can still hydrolyze ATP, and instead of going around looking for a new step, it's decides it's just going to land here, hydrolyzes to ATP. So that has to be included. Right, so there's something remarkable about this. As you increase the temperature, the distribution gets narrower, but the center of the distribution is exactly the same. So that was another clue that started thinking, can we understand this? When I showed the original data to Ron Vail, he looked at this and said, wow, at body temperature, the motor works the way it does. It's supposed to. It doesn't take backward steps. Okay, but again, you have to really know your apparatus and what you can't see and see. So let's say there are 4 nanometer and minus 4 nanometer and 0 steps. And let's do a very simple model, because when this thing steps, it's going to be, going to pull the vesicle, and the vesicle has a viscous drag force in the viscosity of the cell. So we say, well, it's a spring constant. We know the size of the vesicle. There are pretty good estimates about what the viscosity is in the axons. And so that's one equation, the drag force. And we're just going to say, when it takes a step, it exerts a force, and it's pulling this vesicle through this viscous stuff. So that's great, because you make the forces, the spring constant, the drag force, and you can solve that equation. And what you find is it's a statistical equation. So when you see bracket x squared, it's the statistical average of the steps looks like 2 kT over the spring constant times this form here. Very, very nice intuitive form, because you can go to some limits. For very, very short times, you can do an expansion, a Taylor series expansion of the exponential, and what you get is the step size distribution is just twice some diffusion constant. So it looks like pure bounding motion. If you wait a long time, you can think of this vesicle as jiggling around. Remember, it's stepping here, it's attached to a spring, and it's jiggling around. And for a very long time, you explore all the allowable phase space given the spring constant. And you have this limit, that 1/2 kx squared equals 1/2 kt. Okay, so that's the solution. It makes a lot of simple sense. We don't have a spring constant, it's just a parameter. But the remarkable thing about this is, once you have that, the single parameter spring constant actually fits all the data. So for high temperatures, very short stepping times, it's here. A pure Brownian motion is down here. And as you go to longer and longer times, because it's thermally activation, so it takes longer for the PI to leave, that is the rate limiting step, you begin to saturate and explore all of phase space, just the way the equal partition theorem says it's true. So that's encouraging. Simple model quantitatively fits this, but it gives us a parameter we know nothing about yet. Then we go and look at the literature, and there are two general structural states, a combination of crystallography, but most important, the cryo-EM stuff. And after it's made a step, and that jiggly linker arm has swung down, so in this cartoon, the linker is attached over here, AAA one side, it's attached down here, AAA side, and this is the tail. So we draw a little stick figure. We also know the angle between this stalk and the microtubule it's working on is 55 degrees. And when it's in this exploratory state looking around, we also know the structure of that. We know the average bending size of this. It's up, just moving around, really. And we know this angle, and we know this distance. Okay, so here it is. You've got two legs. You're going around. You're stepping around. You start to land. You say, okay, I've finished this step. The thing moves forward. And remember, it's a random walk. But because when it's in the exploratory mode, it's biased forward, this structure tells you that the bias forward, just from the crystallographic data, should be 10.8 nanometers. But because when you encapsulate the other thing has not taken a step, the bias forward should be 10.4 nanometers. But we fit to, it should be 5.4 nanometers, half of the bias forward of the single step. But our fit was 5 nanometers. And believe me, in biology, that's really close. Because we were just taking measured things, and there's little subtleties in how the thing is attached. So I said, that's great. So we have now the structure telling us it's a random walk, bias forward, it's thermally activated, and we understand why the center distribution is where it is. And so that's what I mean by you're beginning to get a quantitative understanding, or at least measuring parameters of what this thing does. Now, we went much further than that. Since you're mostly physicists, I'm not going to tell you the detailed model. It's very, very complicated, but I'm just going to skip that because I want to teach you about some interesting physics. Okay, so the model is out there. It's in a bioarchive. They didn't send it to review. Finally, we got the first quarter of the information in the bioarchive, which is, I have to say, very hard to read. So it got into Nature Chem Bio, and we want to get a couple more publications. But let me talk about non-equilibrium statistical physics, okay? So we have this measurement. We're beginning to see we can make quantitative measurements, what it's doing. We have a proposed model, but once you have a proposed quantitative model, you want to verify it. And so, but before I do that, I want to tell you about what's called, it's a new theorem in statistical physics called the fluctuation theorem. And so let me start with a collision, two molecules colliding, or two atoms colliding, and going forward in time, they bounce back. And you know if you run the movie backward and go like this, you can't tell forward from backward, right? Very, very different. I'm sorry, the same. Time reversal invariance, because the microscopic interactions, E and M, are time reversal invariant. But suppose you drop a little blue ink into a beaker of water, and you wait a while, and the blue ink molecules say, hey, I used to be in this little droplet, but where can I be? I can be anywhere in the beaker of water. And if you wait for a period of time, These blue molecules have, on statistical average, they can be anywhere in this. And so you get this uniform color bleed. You know which way time is going forward when you have this. And we all know, if in physics, that this is a manifestation that the entropy of the system, clear water and a drop of the blue is a certain amount of water, but when the blue molecules are everywhere, there are many more accessible states, so entropy is increased. So that's what things do in a closed system. Either entropy remains the same or it increases. Okay, so that we know, that's from the days of Boltzmann. But there's a theorem that says the following. What if the probability of running the movie forward? Running the movie forward is a blue droplet is now in here and it goes to evenly distributed blue molecules, divided by the probability of running the movie backward, going backward in time. This theorem says it is equal to the change in the entropy divided by Boltzmann's constant. Boltzmann's constant is just a normalization constant. You know, you have as units of energy kT. Temperature should have been measured in joules, but it was measured in Celsius or Kelvin. But it's temperature. So what does this say? You're allowed to go backward in time for brief little instants because the delta S is small. Okay? So this is nice because you say, yeah, time reversal invariance is still there, but it's the law of large numbers that tell you you're going to end up with exponentially large probability of going forward in time, not backward. And this is called the fluctuation theorem. It is not, there's time in here. It's not equilibrium. Equilibrium means you waited for an infinite amount of time, point number one. Point number two, it was discovered numerically accidentally in 1993, not 1883. And then proven heuristically a year later and subsequently made very rigorous. So this is a theorem that actually tells you how entropy is flowing in a quantitative way. Okay, it's a new theorem in statistical physics. Let me give you an example. This was an actual experiment that was done. You take some particles and you drop it and you make sure you get rid of all the air currents and you drop these particles and you watch these particles drop and it's in viscous drag. It's not in equilibrium because the equilibrium of the particles is down at the bottom because there's a constant force pulling it down, gravity, and there's this viscous drag slowing it up, and of course there are fluctuations, Brownian motion. And they started measuring with some high resolution what is happening, and not surprisingly, if you go at high enough resolution because of Brownian motion, sometimes they fall up. Okay? So it's kind of going backward in time, where time is the statistical average, which means they fall down uniformly. So it's just fluctuation. It's not a surprising thing. It agrees with the fluctuation theorem, of course, because it's a theorem. It's now a theorem. Okay. And so there's how the ensemble things, if you go more longer and longer distance. So they're falling up, down. Okay, so now let's go back to our motor. This is not a simple little particle falling in viscous drag air. It's very complicated chemical reactions, and you just take one motor with one vessel and it starts to walk around, and after a certain time, it's over here. This is a statistical statement. You start again with another motor and walks around, and after a little time, it's over here. And so in terms of time, maybe it can walk backward a little bit, maybe it walks forward, it's biased forward. And so how do you reverse time? Well, when you're burning energy, there's so many degrees of freedom that it's really, you don't go backward in time by burning something. This is how biology makes things go forward. There are too many degrees of freedom. But what we can do, so we can't do this in the lab, but it's easy to make a time reverse thing. So for example, you start at the origin and you end up here. What's the time reverse thing? You start over here and you end up back at the origin. Similarly, you start the origin over here, you end up here. Time reversed, you go back to the origin. So, but if you start time equals 0, you just take that trajectory. And so going forward in time is going up like this. Going backward in time is going down like this. So it's just a algebraic construction. Now, the beautiful thing about this is the distribution probability we're measuring are Gaussian. And the wonderful thing about Gaussians is the fluctuations thing says the probability distribution going forward in time divided by the probability distribution going backward in time has to be e to the change in the entropy, but I can take the log of both sides. Taking the log of both sides, meaning I'm adding and subtracting the stuff in the exponents on the left-hand side, and this becomes delta sk. Okay? So that means, and so if you start going forward in time, and these are now measurements, after 10 milliseconds, the distribution goes forward in time of our vesicles. We track vesicles over a long period of time, and because we can track a single vesicle over a long period of time, you have enough statistics to build up distributions. So after 10 milliseconds, this is the distribution. After 40, this is the distribution. After 100, that's the distribution. So it's moving forward, and the distribution is widening. Going reverse in time, after 10 milliseconds, you go backward like here. So it's just arithmetic algebraic distribution. You take the log of these sides, and you find this funny quantity. You do some algebra, and you find this phi quantity is equal to twice the Gaussian distribution is characterized by a mean and a dispersion. Mean is mu in this notation. The dispersion is s squared, is dispersion squared. So that's what the fluctuation theorem tells us. But there's something very important about the fluctuation theorem, because it's talking now about entropy. And once you're talking about entropy, you can start to think about the thermodynamics or statistical physics of this. And so the associated energy or heat associated with this T delta S change in entropy with a drag force, you can just write down what it is. And you do some more arithmetic. And what you find is you end up, right, we're measuring going forward in time, and you find that, let's talk about the enthalpic energy. ATP lands on, or a couple of ATPs land in this, they hydrolyze, they burn up energy, but then after a full cycle, guess what? You're starting new ATP. So our system is not the supply of ATP, our system is just the motor and the vesicle. And in that, since you're going full cycle on the ATP, the net enthalpy is 0. So that goes to 0. And then the rest of it's just the external work. Well, the external work has two parts. One is the work you see. You're dragging this vesicle through a viscous medium. That's force times distance. And you know the viscosity, you know the distance, so you know that. And the other is the internal friction of the motor, which we don't measure. So what we have is actually an inequality that you do this arithmetic and you get this quantity. Phi is measured. Phi is a measured quantity. It's twice the mean displacement divided by s squared, dispersion squared, okay? So that's a measured quantity. We look at 37 degrees, we convert to Kelvin, we find this force has to be less than 0.43 piconewtons. That's what the fluctuation theorem tells us, and it's a theorem. But there's another way of going there. We know the size of the vesicle. We have good estimates of the viscosity. They actually measure the viscosity by a nanoparticle in the soup and watch how it diffuses. So we know this. We can figure out what after a certain time, it's walked a certain distance. So you drag this thing. The other is a measurement. We're making the measurement. So what's going on? Let's suspend belief and say maybe the temperature is really not 310 Kelvin. And if we allow to say there's an effective temperature is not 310 Kelvin, that effective temperature is about 29 times the temperature you made the measurement. Now, so then you're saying, what do you mean 29 times? At that temperature, everything's ionized. And this is in a soup of at whatever temperature you do. So that means we have to examine what temperature means. Okay, so far, some of you are with me, please say yes. Okay, so now I'm going to do a little history. And way back in 1931, Lars Sauger made a conjecture, and what he was trying to do was trying to consider small perturbations out of thermal equilibrium. Can we begin to discuss non-thermal equilibrium procedures? And his conjecture was, suppose you have a system in thermal equilibrium. We have a theorem called the fluctuation dissipation theorem. It was first finessed by Einstein, but it was actually first conjectured by people at Bell Labs and proven by some theoretical physicists in the 50s. And it said there's an intimate connection between fluctuations, like Brownian motion, and dissipation, in this case, viscous drag. It was conjectured by people at Bell Laboratories who discovered that there's a thermal noise called Johnson noise, and there's a resistance. And so the Bell Labs people said, maybe there's an intimate connection between the fluctuations in voltage and the resistance, which is the dissipation. And in Einstein's Brownian motion, that's very clear. It's Brownian, jiggles on molecules on both sides of the fluctuations and viscous drag. And so what Onsager said, these are the equations, but it's not really important. What he says is consider a system slightly out of thermal equilibrium. So do a Taylor series expansion out of thermal equilibrium. He said its approach back to thermal equilibrium would be related to the fluctuation dissipation theorem. The fluctuations, for example, are fluctuations in space, so it's a correlation function. And the dissipation is, force and distance, drag, okay? Much later, he gets the Nobel Prize for this because of the first attempt to try to think of non-equilibrium statistical physics. In hindsight, it had to be true. There's no way it could not be true because consider you're looking at a system in thermal equilibrium, but you just take a snapshot and begin the movie when it's 3 sigma away. Its fluctuation is correlated to dissipation. You don't actually know that it was purposely put out of thermal equilibrium. So it had to be true. And it turned out analytically to be proven to be true. And so that's the Lars-Onsager conjecture. But the amazing thing about this is it was later to be proved not only for small perturbations out of equilibrium, it's also true for very large departures from equilibrium. And in this formulation, you can define an effective temperature. Okay, it's when this quantity delta chi is this essentially distance over the force times the correlation function reaches a constant. Okay, now I'm a simple physicist. I read this stuff. I read it again. I read it again. Finally, I read it enough times I could remember all the steps and say, okay, I got it. But being a simple-minded physicist, I want to reduce it to something that's so intuitive it's like the back of my hand. So I said, let's start with the fundamentals. Fundamentals are F equals ma. In a world of Brownian motion, the ma, mass times acceleration, it has an external force. It has the viscous drag force. And it's got this funny jiggly term, which is the Brownian motion term. And so I'm going to turn the motor force into an average motor force as it's stepping. But remember, when it steps, it's stochastic. How far it steps is also stochastic. But you can, over a period of time, you can make an average force. And then the stochastic nature comes as a fluctuating term. Okay? And then you do something really dumb. you change the frame of reference. Now you go in the frame of reference of the vesicle and motor, and when you go to the frame of reference of the vesicle and motor, you get mass times acceleration is equal to gamma times this change of reference times 2 jiggly terms. Okay, and you look at the jiggly term, and if you look at a time resolution, that means that you're averaging over at least a couple of steps, so you've lost correlation of the history. It looks exactly the same as our old friend Brownian motion. Okay, so you just have to look after a couple of steps, and so when you see after a period of time you have a distribution, and another period of time it's a wider distribution, and another period of time the distribution is even wider. Brownian motion says when you start at 0 and you diffuse, so how fast is it diffusing? And lo and behold, the diffusion rate is described by this effective temperature. That's all it is. So what that really means, and now why is it that's such a big deal, and why is it so obvious once you see what's going on? You got this big sphere, 150 nanometers, 100 nanometers in radius. Brownian motion is jiggling. But it's a big thing. It's not a little molecule, it's a big thing. And so it doesn't really move that much. And the bigger it is, bowling balls don't have much observable Brownian motion. Little molecules can jiggle a lot. But then you got this motor. And in the frame of reference, it's stepping forward, it's stepping back. The size of the steps is stochastic, but it's really powerful. So you got a fluctuating term that is able to move the bowling ball easily. And because it's so powerful, the Brownian motion part is irrelevant. And so the widening fluctuation is due to the fact that you got a big molecular motor with a lot of energy that makes it jiggle. Okay? So that's all effective temperature means. It's a big disappointment. It's kind of cool, but there's more. Okay? So far with me. We got a fluctuation theorem proven rigorously in around year 2000, 20 years ago, 25 years ago. And we have this system that appears to be out of effective equilibrium, a la the formalism that was started by Lars Onsager. Now, I'm going to tell you remind you a little bit more about statistical physics. And this is a Maxwell demon. So the idea of the Maxwell demon, just to remind you, you got a bunch of hot molecules and cold molecules. You got a little smart demon, and it looks at that red molecule going to the right, and it opens a little trap door and allows the red molecule, the hot one, to go in here, the one with higher velocity. They're bouncing around, and the cold ones, it allows to go this way. So it decides whether it's hot or cold, and after a while, you've got hot and cold. And so a paradox that Maxwell was saying is, so what's going on here? You're allowed to take something and you can turn it to hot and cold, and once you have a temperature difference, you can do work. And Szilard's version of Maxwell demon strips it to its bare essence. So he said, suppose you got in a little box one molecule, and then you put a door on it, and so it's either on the right-hand side or it's left-hand side. But if you know it's on the, let's say, left-hand side, and it's bouncing around, after many, many bounces, it pushes the piston this way, okay? So you can extract work, and it does that, and therefore you've got work, but that's just one thing. Now what do? You take away the door and slam the door again. You say, is it on the right-hand side or left-hand side? Oh, it's on the right-hand side now. The piston's going to push this way. So by knowing which side it's on, either right or left-hand side, you can actually extract work at the same temperature, which thermodynamics says you can't do that. Perpetual motion machine. So that's the essence of the Maxwell-Demon paradox. Okay? It violates the second law of thermodynamics. So just hold the thought, and then let me tell you about other great things that happen at Bell Labs. Many great things happen at Bell Labs. And in the 30s and 40s, Claude Shannon was interested in thinking about how information gets transmitted across telephone wires. And he invented an information entropy, which is equal to the sum. entropy is defined as Boltzmann's constant times the log of all the accessible states. But another formulation, if you count up all the number of accessible states, whether it's finite or infinite, it's the probability of being in one accessible state, the logarithm, and you sum up all the accessible states. So that's normal entropy. Shannon says, I can make the same thing. Let me give you a really simple example. If you have one bit, it could be a 0 or 1. And those are the accessible states. So the entropy of one bit, if you look at his formula, it's minus 1/2. The probability is 1/2 of being, if the equal probability of being up or down, 1/2 times the log of 1/2, and plus 1/2 the log of 1/2. And so you do this, and you do log base 2 to make the arithmetic easier. So the entropy of one bit is 1. Then the reason he wanted it in log was he wanted, a string of bits, so the becomes additive. So consider a simple example. Suppose you have a coin, flip it, heads or tails. It's an equal probability of being heads or tails, so the entropy is 1. But then you look, it's either heads or tails. Then the probability is 1 of being head or tails, and that entropy is 0. And so in the act of looking, you've changed the entropy from a 1 bit, from a 1 to a 0. Okay? Because not all heads or tails are equally likely, but you've looked. And so the act of looking, you've actually changed something thermodynamically. And so here's the big intellectual leap. There's a thermodynamic entropy, the way Boltzmann taught it to us, the same form with a Boltzmann's constant in front of it. So put a Boltzmann's constant in front of Shannon's entropy, and the total entropy is just the sum of the two. And so if you say the real entropy is both the information entropy and the old-fashioned thermodynamic entropy we knew about, it generalizes the second law of thermodynamics. So sounds kind of goofy, but it took a while to make this bold leap. To say, put it in equal footing. And now, you notice that we're not going to look in the details in this Szilard version. You got a box, you put a thing in, how do you find out what's left or right? We're only going to look at a limit. What's the, in looking, you're changing entropy. You can look as well as you want. It's like uncertainty principle of delta x, delta p, greater than or equal to h bar. I don't care how you look at it, you're going to be limited by this. And so in this generalization, you can do this. So that was a big leap. And about a little while later, some physicists at MIT were thinking and asking the question, what is the minimum energy you need to do a computation? And it's like when Carnot talked about heat engines, he said, what's the most energy you can extract from temperature T1 and T2? And you do this. So Carnot invented what's called a frictionless engine. Frictionless means you go very slowly, there's no convection, there's no nothing, so it becomes quasi-reversible. So reversible transition. So these guys, Landauer says, can you do this? And they found that you can actually do a computation and to make it reversible, and you don't need any energy. You're doing a computation at temperature T, it can be completely reversible, no energy. But they found that reading out the state of the system, whether you're in a memory, so when you have a memory, it can be either 1 or 0. You look, it's 1 or 0, you've changed the entropy, and that's irreversible. And that you couldn't avoid. The computation is actually reversible, but the reading of the memory is irreversible. It's not a big amount of energy. It's 10 to the minus 21 joules. It's been verified. A colleague of mine at Bell Labs, when I was in the department, he was in my department, he's now a professor at Berkeley, electrical engineering. He figured out a way to gently look at a spin, and these are temperatures, and the black dots are the Landauer limit, and his dots are experimental data, and you see as you go up to 400 Kelvin, you're actually reaching the Landauer limit. So this is very cool because you've quantified a net unit of entropy, and it's now measurable. It's not an abstract thing. Cattell wrote in one of his, Charlie Cattell in his condensed matter physics, said, entropy is just a fictitious thing. No, it's measurable. Okay, so that's also very cool. So entropy is measurable. We've unified information entropy with thermodynamic entropy, and now I'm going to, in the last remaining, I'm a little bit over, so in the last remaining half hour, just kidding, I want to tell you very quickly how dynein exploits thermal fluctuations. One of the great physicists of the 20th century, Erwin Schrödinger, we're in the 100th anniversary of the founding of quantum mechanics, of which he played a crucial role. And he said, he started getting interested in biology, said that, I think with biology, information is the key. Maybe there's some thing that encodes information. Maybe it could be a long linear thing, before DNA was there. But he also said in this set of lectures he gave in Dublin, Ireland, from all we've learned from the structure of living matter, we must be prepared to find it working in a manner that cannot be reduced to ordinary laws of physics. But it's not because there's any new force, but because the construction is different from anything we have yet tested in the physical laboratory. And if you think on a nanoscale, micro-nanoscale, if Newton were the size of a bacteria but still had his brain, his laws of motion would be very different. Object in motion quickly comes to rest. So there's no inertia. And there's this crazy Brownian motion, like the hand of God just shaking you for no good reason. And so it's not F equals ma, it's F equals mv, where the proportionality constant in the case of a viscous drag is just the Stokes drag. Okay, so we make macroscopic motors to get rid of friction, and it's great, but these motors have to work in this environment where the laws of motion are radically different. Now, when people think of what motors look like, they're still thinking of the macro world. And in this arm, this is a cartoon of dynein. Remember, it's this linker arm. This is the vesicle, and there are various stages. And here it's looking around, deciding where to take a next step. It plants its foot down, and then they invoke what's called a power stroke, and something makes this swing down to this position. And when it swings down, it's connected to your vesicle, and the vesicle moves. Okay? So that's the language of molecular motors. They always invoke what's called a power stroke. So being a newbie in this, I said, okay, you're the experts in dynein. Where's the power stroke coming? I said, well, we really don't know, but it must be there. Okay. But then being a physicist, I said, oh, suppose there is a power stroke, and you suddenly pull down, and then this thing's attached to a spring. So let's say, okay, it's attached to a spring, the spring constant, and it comes down, differential equation. And what you find is the relaxation of the vesicle to the new position, the time constant is independent of the size of the step. Okay? If there is a power stroke that goes and just suddenly stroke, it does this, you have a very quantitative prediction. But let's do a cartoon. Suppose here's the vesicle, and it's now in this position. ATP lands, and so it's maybe in this position, or maybe it wants to take a bigger step, that linker arm is bent more, or even a bigger step, the linker arm is bent more, okay? And so finally it straightens up, and when it straightens up, the vesicle moves forward. But what makes it straighten up? You can invoke a power stroke even though you don't know where it comes from, but suppose you say, what if there is no power stroke? What if you look at these various positions, and you think of the equilibrium as somewhere in the middle, and it's got to do a Brownian motion fluctuation to get over here to where it is allowed to redock, and the chemistry of allowing to redock is understood. So the hydrolysis of ATP and things like that allow to undock and redock, but there's no gross motion, and these things can be stepping by 10 to 20 nanometers. And so it becomes a diffusion question. If you have a big step, you have to diffuse over here. If a small step, you diffuse over here. But in the diffusion and it locks in place, you've got a little cock spring, okay? So that's the model of what we do. And so that means if if there's no power stroke, but it depends on diffusion, then the time it takes to go from one position to another, that transition time, would be a function of how big the step is. And it turns out there was a very brilliant experiment done a long time ago by Sunny Shi and Xiaolin Nan, who then became my postdoc, who's now doing some cancer research. But they wanted to demonstrate the method, and they found, and they only had 3 data points, they found that if it takes a big step, 20 nanometers, the time it took was about 500 microseconds, but a short step, it took 120 microseconds, okay? So 3 data points suggesting, but they didn't pursue it, and they said, well, we may come back to this later, and then it was all dropped. Now, because of COVID, we've been thinking about this, said, this is cool. We have a definite prediction because it's diffusion, but you got to get a lot of statistics because it's a statistical prediction. So you have to measure not only where it's going, how big a step is, but the time of making the step. And so what we did was we followed them, except there were two crucial differences. Number one, we had something we were going to test. Number two, they used gold nanoparticles, and they were excited in the green, and so you're limited by when we cook the cell. And it turns out you can buy gold shell nanoparticles, which shift the resonance to the infrared, and you can put in lots more power. And so what do? We take this gold nanoshell, it's like our rare earth nanoparticles, we put on some molecules that fool the cell and say, take me back to your cell body, puts on all the motor proteins, and off we go. But now we're scattering light. This work was started by a physicist who used to work on atomic clocks, got 20 digits worth of precision on atomic clocks, and it's being finished by Jeremy Axelrod, who was a PhD student by Holger Muller. Holger Muller was a postdoc of mine who used animated photometry to measure the fine structure constant. And he just started in March. But Holger was a graduate student of Achim Peters, who was a graduate student of mine. So, and Achim Kenyao and I used animated photometry, measured the acceleration of gravity at very high precision. So I'm getting second and third generation people, which is kind of cool. So the idea is very simple. You have your nanoparticle, you put them on here, they get endocytosed, motors are put on, they're chucking back. You take a superluminescent diode. You don't want a laser because lasers give you speckle. So you want a broadband incoherent thing. You use what's called dark field microscopy. You come in and illuminate here, but you look in a cone over here so you don't get any specularly reflected light. And you do something very simple. You use a blob of light, and suppose the blob of light moves. We're no longer going to use a camera, because if you use a camera, the speed of reading out the camera is a problem. And the high-speed cameras have a lot of noise. So we're going to say, we just want to see the blob of light. And if the blob of light is on a split field photodetector, and you go over here, this creates a positive voltage, this creates a negative voltage, and in an op-amp, you get a voltage. You can do this with engineer a little bit better, so you have a knife edge. You put into 2 fast photodetectors that go to this differential amplifier, and you digitize it, and you set the set point so it's in the middle, and in the middle it's linear. And that's what the apparatus looks like. We finally got it up and running. Again, it was pretty good because this guy's pretty good. It was March of 2024. And this is the noise spectrum going to higher and higher frequencies. This is the Brownian motion, that's the instrument noise. These are orders of magnitude. So well below Brownian motion, the instrument noise, okay? And this is a live neuron at 22 degrees. And so he made the electronics himself. You can buy a double bounce mixer and everything, but we can do a factor of 5 or 10 better than anything you can buy commercially. And so you can track this, and then you can look at it, because it's in the infrared at 800, 850 nanometers, you can watch it forever. You're not heating up anything, there's no reactive ion species. And And if you put it in a filter and ask for 1.125 microsecond resolution, okay, over here, you have extreme sensitivity. You have angstrom sensitivity with 1 microsecond time resolution. And what do we see? The minimum step of a single two-legged motor, the minimum step is 8 nanometers. Could be 16. If it's 16, this thing moves 8. If it's 8, this thing moves 4. Okay, it moves backward. And these are the minimum step of a single motor. And you look at this, it's still the single motors. But unfortunately, there's much more going on now. And we're puzzled, now that you've improved the time and space resolution by a couple of order of magnitude, both of them simultaneously, all of a sudden you're seeing things that we couldn't see before and weren't seen by earlier people. So now we're getting into hog heaven because you're beginning to see, and so how do you make sense of it? This is a single step, and you see it jiggling around, there's transient stuff, and it gets to be really juicy as a scientist and say, new instrument, new couple orders of magnitude, Yogi Berra, new set of eyes, you get to see new stuff. So in summary, the understanding of the literature and the data analysis was done by Sam Peng and I in those long COVID 2 years. They were assisted in the early stage by Yongheng Zhang and Quan Lu, who are now professors at Fudan University. Ed Marty joined. He was the clock guy. He then got an offer from Washington University of St. Louis, but somehow I can't get rid of him. I said, take the job. And he's, anyway, and I can't afford him. And Jeremy Axelrod. Ben Shao was a postdoc when the work started, studying retrograde motion and nerve growth factor. She's now a professor in the chemistry department at Stanford. Tom Sudoff supplied us with neurons that are induced to be human neurons from fibroblast cells. And at that time, Alan Wong, we have another person. Of course, I couldn't get any funding because they said, you can't do any of this stuff. And besides, you don't even know biology. So we got a little seed grant, finally more grant. And after we got preliminary results, we can actually get NIH funding. So that's the usual story when I've changed fields. But I'm very excited because the data is saying we were expecting to see something neat and clean in our model, but we're seeing some of that, but we're seeing something else. And that's going to be very cool. We're going to try this not only with dynein, but with kinesin. I deeply believe, but it's a religious statement, that these things work on fluctuations. They do not work on power strokes, because that's the way Schrödinger was right. He didn't understand what really mattered, but he was right. These motors work by naming new laws of physics, but it's something we're just not used to because we don't live in the micro nano world. So with that, I'll stop.